So if you go on the website, currently you would end up with like some documentation that shows like the basic operations using like GIFs. And the most advanced part of it is like this interactive pathway view, where metabolic pathway visualizations are coupled to like mathematical simulations that are running in the background on servers that we have. And then you can just like basically edit. You can remove genes, you can remove reactions, you can add reactions, you can change the medium, like add a different carbon source, remove a carbon source. So all the things that when I started here at CFB, was basically doing in Python code, you can basically just do now by clicking around on a map.
Cool.
And doing basically also usability tests with like biologists, they find it like pretty intuitive and actually can find their way around without a lot of documentation or like tutorials or and so on. So here, for example, I had to remove both genes for this reaction because they're like two isozymes, and if I remove only one of them, then it's still functioning. So it's also like has a good relation of all the components in the cell. And I can always like undo operations, like add the gene back in, then the reaction runs again. Here I'm like changing the carbon source from, like currently it's basically taking up glucose as the sugar. So I'm removing glucose. Then basically the cell is dead, there's no carbon, right? So I'm basically adding pyruvate instead as a carbon source. And then you basically get very different distribution of metabolic fluxes. So that's basically the very manual part of the platform where people, if they have a good idea about a design, they can actually question a model. What would the model say about it? Because the model has a much larger scope than one can have in their head, right? Another aspect is that you can add, you can integrate experimental data. For that you have to log in first because you need to be able to upload data. So here we're going to integrate a data set that came from a carbon labeling experiment where basically isotopically labeled glucose was fed. So you can resolve like some fluxes inside the cell. Just going to load this data set. And then the important part is this looks very easy, but it's actually very hard on like in the background because it has to apply like all the conditions, like what is the genotype of that strain, what was the medium that was used, what are the individual data points, and so on. And all of this basically automatically, adjusts the model to reflect the experimental reality. And then basically predicts based on those data points. It gets more accurate predictions about the rest. So you see all the reactions that have a glow are the actual experimental data points, and everything that is not glowing is basically inferred based on the data and just predicted. So you get actually more out of this than just the data that you had initially. And the other aspect is that we want to have like strain design should be really push button. So the most minimal input that you can give to this design pipeline is just the organism that you want to use to engineer and the product, and it will basically just predict what heterologous genes do I need to express in order to get a functioning pathway, and not just like one, but all alternative pathways that you could potentially find. So here it found like one step, and it also connects the reactions that are predicted for the heterologous pathway to like the sequence level, and that is actually taking advantage of like the work that has been done at EMBL. Like here, if I click on the GMGC link here, it will basically redirect me to a site that has like the 10,000s of like metagenomic sequences that have been, from the human gut, environmental samples, the ocean, whatever. So you could like find like gene targets that you could synthesize in order and then like basically introduce into the strain. And if you select like one of those designs, you can actually share it with this interactive pathway map. It will automatically add those reactions that have not been in the model because the model reflects kind of the wild type. So if you add those heterologous reactions, then I could, for example, say, what is the maximum flux through this reaction? And then answer questions, okay, what is the maximum production rate or the maximum yield that I could get based on the carbon source that is set? But I could also change the carbon source and, ask additional questions, allowing me to kind of prioritize out of the hundreds of designs that this pipeline spit out, like what is the thing that I should go and build in the lab?